raise your hand if you don't want to, but okay, yeah. The interesting thing here is that most people said it was true by about 72, almost 73%, which is kind of astounding because anybody that's actually in origin of life science would say, well, obviously we haven't done it in the lab, even if they think it's possible or they're working toward it. Everyone knows that no one's come remotely close to that. They also asked in the survey, do you think we've ever created frogs from non-life? I forget the percentage, but it was a surprising number of people that also thought that was something that science had done as well. So how do we get in this state where the public is so unaware of the science related to the origin of life? There's this big disconnect. And part of the problem is the media, they'll overblow. Oh, we've made this discovery about origin of life. We now know how it originated on Earth or something. Or there was a case where Craig Venter, a prominent scientist, did sort of a genome transplant into another organism that some kind of mislabeled as creating life in the lab, but it really was far from that. But the interesting thing too here is that it's not like they were just asking a lot of uneducated people here. 80% of the people that responded to the survey had degrees from college. So it wasn't like a totally ignorant populace that they asked that question to. So I'm going to now give you 12 different aspects to the origin of life that are challenges. The way I like to phrase it is maybe some of these are insurmountable or not, but they form a pretty strong cumulative case because each challenge I don't think really has been met by plausible science at this point to explain how life could originate from non-life based on these challenges. Maybe some of them will, be passed, but at this point, I don't think any of them have, they're challenges that are completely unmet based on current science, is what I'm trying to say. So the first one to discuss is cellular complexity. So one of the ways in which origin of life scientists approach the problem is to start with an existing life form, a simple bacterium or something, and do genetic knockout experiments. Okay, what happens if we get rid of these genes? Can it live? And so they try to just get rid of as much as possible where it's still alive as a way of finding out, well, what is the simplest possible life form? Okay, so that's kind of interesting, right? And what they found is that it's actually still quite complicated. They found that the simplest one they could find had about 450 genes. So that's a lot of complexity because in each of those genes there's at least an average of about 500 base pairs. These would be nucleotide code, letters and then nucleotides, letters corresponding to nucleotides for a genetic code. Okay, so you multiply that together and that's a lot of information, right? A lot of things that have to be just right to even just specify the blueprint for the organism. And actually that was a parasite. That was something which depended on other life forms to live. Okay, so the 450 was based on that. So the most generous or the most optimistic evidence of how, or estimate rather, of how much you could reduce the size of the genome for the simplest possible life that I've seen is about 200 genes. Most would say that it needs more than that. But even there, that's 100,000 base pairs in the genome that you have to get just right, just to code for that organism. So that makes it really hard, right, to get that first life form. And I think a lot of scientists recognize this. Lynn Margulis, National Academy of Sciences scientist, she said to go from a bacterium to people is less of a step than to go from a mixture of amino acids to a bacterium. So, part of the evolutionary story is that everything, has this common ancestry going all the way back to a bacteria. We talked about that a little bit last week, but as hard as that is to believe, she's saying it's even more improbable to get the first bacterium from non-life. So this is recognized as being a significant challenge. Stuart Kaufman, he's an origin of life researcher, and he's attacked one of the theories for origin of life that we'll talk about more later, which is called RNA world. This is the theory that okay, yeah, the first cell, that seems too hard. Maybe there's some proto-life that could have evolved through some natural selection, some kind of chemical evolution. And for reasons we'll get to later as well, some people think that was possibly RNA. And so he's attacking even that and just saying that he finds that to be ridiculous because all living things seem to have a minimal complexity below which it's impossible to go. And so he's kind of tying into this cellular complexity theme that you see on this slide that as far as we know, all life forms, you have to have a membrane to protect everything inside the cell from the external world, to have some kind of homeostasis. And you need to have all this kind of information. You've got to have things like ribosomes that can make proteins. So people that try to do away with that, to some researchers, at least in the community, they're very skeptical. Could you really have something like that is a self-sustaining life form that could evolve, which is what naturalism would demand? But let's take a step back. Let's start at sort of the very beginning of what most people point to in trying to craft some kind of narrative about origin of life. So how many of you guys have heard in some class probably something about like a primordial soup where some organisms, or maybe at first it was just some organic compounds that were formed, and then they kind of built up over time and maybe had some kind of life form come about within this kind of a organic soup. But actually within the origin of life community, there's a lot of pessimism about the primordial soup even. For one thing, organic soup, the fact that it would be organic, is a bit of a challenge because carbon is not, even though obviously all living things are made up of carbon as one of the key elements, but it's only about 1 part in 1,000 in the Earth's crust. And also nitrogen is one of the other main chemicals of life. It's even rarer, 20 parts per million in the Earth's crust, even though there's a lot in the atmosphere, admittedly. But in order to get some kind of high concentrations of organic molecules in some kind of soup, some kind of aqueous solution, for a lot of people that know about the early Earth, is deemed to be something which is fairly improbable. Kind of blocking your view there, aren't I?
I can actually cheat and see it.
Okay. So everything we know about geology, geological depletion prevents the accumulation of these organic compounds. They're not. It's really everything all the examples of large stores of organic things that we know about were always caused by life. There's not any known processes that would sort of segregate, okay, let's get these carbon. Life does it, right? But we're talking about what happens before you get the first life. So there's no known geologic processes that are going to like isolate carbon and let it build up in concentration somewhere. And as some like Robert Shapiro, one of the top origin of life researchers as well, have pointed out, really what you get when you start even if you have a soup, it really goes towards just producing like something that's basically like tar. You keep having these you have these chemical reactions that just build up, and you keep connecting these organic compounds together to form tar. So we don't have as good a starting place as you might be led to believe by a few textbook kind of diagrams. But let's talk about the other thing you see a lot in textbooks. How many of y'all have seen this? The Miller-Urey experiment pictured there. This was from the 1950s, where Stanley Miller, when he was a graduate student working under Urey at the University of Chicago, put together what did deserve, I think, a Nobel Prize. It did some good work to show that you could form amino acids from something which is relatively hands-off kind of setup, where he just basically, he tried to simulate actually he admitted later that he chose the atmosphere based on what he knew would work to give you amino acids. But at the time that he did it, there was nothing wrong with that because they just really didn't know. But later, it came under scrutiny because he assumed what's called a reducing environment. But a lot of people think there probably was oxygen in the environment, maybe not nearly as much as there is now, because plants release oxygen. But there are geological processes, and just having water in the upper atmosphere, it turns out the rays of the sun will split it, and you'll end up forming some oxygen from doing that. So anyway, yeah, the Miller-Urey experiment was able to produce some amino acids. They kind of segregated out the amino acids from the rest of it. They added a spark. But there's some issues with it that we'll get to in the bullets here. But it was a significant boost forward. It led to a lot of people researching origin of life. A lot of money started going into the field, which is good, right? We want that. We want people to find out what the status is. And so that was a significant experiment. So really what you're looking for life, though, it's more than amino acids. If you want to get to the building, really we're talking about the building blocks of the building blocks. You have in all organisms, you have proteins. So yeah, it's not just things you eat, right? But proteins have a structural aspect, and a lot of proteins are enzymes. So they're catalysts to drive chemical reactions. Without those, the processes that you need go just sometimes million or billion times too slow. It's very important to life to have these kind of catalysts. And then you also have lipids, which are like fats, polysaccharides, or some kind of carbohydrates that you need for life. So yeah, those together form about 90% of the dry weight of a bacterium. So a key question is like, well, how hard is it to get those building blocks? But yeah, part of the challenge of this, the great thing about organic chemistry, right, is you can build these long chain molecules using carbon because of its very specialized nature, which, by the way, one of the fine-tuning aspects is that it's very improbable that we should even have carbon and oxygen in the universe. If it weren't for just a certain kind of nucleosynthesis in stars, they wouldn't be able to produce both. We would have never had any carbon, or we would have never had any oxygen in the universe to begin with. So that's kind of cool. But anyway, so yeah, you're trying to form relatively large molecules. It's much more probable to get smaller ones than it is these larger ones. The vast majority of these did not. No, people don't think these were really just like lying around on the early Earth. It's more the case that origin of life researchers have tried all kind of ways to get some level for some of these. They've made some progress. They have some theories for how some of them are formed. But as you'll see, there's problems really with them. So the Miller-Urey originally was just amino acids. Some people have later done it again and tried to show you could form some RNA from it as well. But yeah, when they've redone the experiment with what we now think was actually the atmosphere on the early Earth, you don't get these compounds at all with just modest amounts of oxygen. And by the way, if you don't have any oxygen, that means you don't have any ozone, and therefore the UV rays are rather a problem for splitting up these organic molecules. It's hard to form large polymers. I mean, your body's constantly repairing DNA just from the damage the sun does. But we have these sophisticated repair mechanisms built into us that would not be available on the early Earth. So as Robert Shapiro says, let's sum up. The experiment performed by Miller yielded tar as the most abundant product. And he said there's about 50 of these small building blocks, and only about 2 of these 50 occurred in the Miller-Urey experiment. So it's actually quite modest. And Fuzz Rana actually has talked about how when he went to an Origin of Life conference before, and Stanley Miller was there, he was like kind of segregated off, and it's like people just don't think that's really relevant to the origin of life. Most of them within the Origin of Life community, even though it's still seen as like, yeah, it's somewhat of a milestone, it's an interesting experiment. I'm not saying it shouldn't be in textbooks, but you have to realize just really how modest that is toward getting you life. I mean, if it gets you a few amino acids, and then you've got to chain those together to form proteins, and then you also need these other components, you're still actually pretty far from a self-sustained life form that could evolve. Yeah?
Do you happen to know what some examples of those 50 organic compounds that are called building blocks?
Well, you would have, you know, the adenine, guanine, cytosine, thymine, uracil. You would have, there's 20 sort of canonical amino acids.
Yeah. And each of those count separately.
Yeah.
made.
Okay, there you go. Yeah.
Because there were like, it was 12 amino acids that were made, right? Am I right about that, or was it less?
For Miller-Urey,
In
or
the Miller-Urey experiment, there were like.
the original one, he's claiming, and this guy's an expert, that they only got like 2 of them.
Okay.
But I've certainly read some things where they claim more. But see, a lot of times they're going back, tweaking the experiment. People have found ways to get more, but still others have argued like, well, you're still not really being realistic because you don't have oxygen there and things like that. Yeah.
You said that there were later attempts done that actually yielded RNA.
Yeah, I was reading about one of those this week. Yeah, that is an interesting one. Now, the problem with all of these that we'll get to later is that none of them are giving you. We'll get to there's a whole slide on this. Like, really, what they're giving you, it would never be enough until you solve this other problem that relates to the handedness of the molecules or chirality. So that's a huge problem. No one's ever done any prebiotically relevant experiment to give you homochiral compounds of the type you need to actually do anything biologically. So here it is. That's a good segue. I forgot that. I think I did switch the order of these because it fit better. So most of these building blocks, there's only like one of the amino acids that's not chiral, glycine. There's no mirror image difference. But all the other ones, it's like the same as a right-handed or left-handed molecule. You can have they're basically the same. They have the same chemical and physical properties, even though there's these 2 isomers, these 2 forms of the same molecule. But life only uses one of the 2 forms. So why is that? You might think, well, it doesn't matter. We don't need to do that now. Go ahead, Caitlin. You want to explain it? Oh, an L. Gotcha. So the reason it's important is like if you're forming a protein from amino acids, then you want to use the left-handed. I guess I could advance this so you can see, make sure I get it right also. Anyway, yeah, so you're having to use the same form as you chain them together or the shape of the molecule changes. So if you want a protein to be used as an enzyme, it's got to have a very particular shape because what it's actually doing is helping bring together reactants and lowering the activation energy so that a chemical reaction will proceed more quickly. And so if you change just one of these from a right-handed to a left-handed form and try to build a protein, I mean, you can make the polypeptide bonds and form something, but it's not going to have the same function as if they were all homochiral, if they were all the left-handed amino acids. And so it's really not something debated within the community. People will admit this is a huge problem for which there's no solution, but too often it's ignored. Not all researchers doing some of the publications will always even admit this, or like they're trying to show progress within other constraints, and they're not even trying to address this huge challenge.
I just want to point out that it's just funny that there's like 2 forms of a problem like this, because obviously there's this you're talking about. At least to me, physics-wise, this sounds a whole lot like the matter-antimatter problem with the Big Bang.
Yeah.
The fact that you have to, you should expect that it's 50-50 to come out, but
Yeah.
apparently there's this imbalance of matter that we still don't know exactly how to
Yeah, it
handle.
is pretty analogous to that.
Yeah, it's interesting.
Because almost all known physics processes, you're going to get them in equal matter and antimatter, equal numbers. And it's similar here where unless you start off with a bias for one or the other, almost everything you do keeps them equal. There are a few things like, oh, well, what if we had some stuff on a meteor and there's circularly polarized light? It could selectively destruct one of the 2. But it's usually like a modest amount, and then you still have to deliver that to Earth, right? And what you really want is like 100%. And remember, like most proteins are not short, right? I mean, a really, 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 really short one is like 50 amino acids. 150 is even a short one. A lot of them could be hundreds of amino acids long. So to do something useful and to get 100 out of 100 or whatever the length, all with the same handedness, that's a huge challenge. I mean, you can take 2 to that power, right? And that gives you the probability. All right. So yeah, here's one of the origin of life researchers commenting on this, saying, all speculation on the origin of life by chance cannot survive the first criterion of life. Proteins are left-handed, sugars and DNA and RNA are right-handed. And so it remains unsolved to this day. And it seems that a lot of the attempts to solve the problem have resulted in showing that it's a bigger problem than originally realized. For example, William Bonner spent 25 years looking for a terrestrial mechanism for homochirality. So he's saying, yeah, trying to get all the same form left-handed amino acids or whatever. And he said, trying to investigate them, didn't find any supporting evidence. So he says terrestrial explanations are impotent or non-viable. But it also turns out it's really hard to assume you can get that from outer space as well. And obviously the magnitude, like if you're going to form life, you need a lot of these compounds because you have to get lucky and sequence them in the right ways. And so if you're just getting a few meteors with a slight preponderance of one form or the other, it doesn't really get you that far towards what you ultimately need. All right, so even, I mean, you can almost grant each step of the way here and show that there's still a huge problem. So let's say that somehow you can form a bunch of left-handed amino acids on the early Earth through some prebiotic problem. You still have a huge problem trying to construct proteins from them, right? Because it turns out most ways of arranging amino acids do not lead to functional proteins. And this has been studied pretty extensively at this point. One of the prominent researchers is Doug Axe at Cambridge. He did this work at Cambridge University. It's published in the peer-reviewed journals. It's not just him, though. You can find Robert Sauer at MIT has found similar numbers of improbability. But he says that, he says, look, if in his studies, the ratio of functional to non-functional amino acid sequences was 10 to the negative 77th power. So in other words, for every one functional amino acid sequence, there was 10 or 1 with 77 zeros after it, non-functional ones. So you can see how it's really hard to randomly find the right amino acid sequences to form a protein. Now maybe he's off some, but even if you're off a few orders of magnitude here, it still looks way, way, way too improbable. He even said that in his studies that, and this was all in this peer-reviewed article, and like I said, there's other ones as well, but one of the key problems is that just getting a stable folded protein, what you generally want with proteins is for them to fold over into a 3D shape to be able to be an enzyme, have some kind of lock and key, hand and glove fit to help speed up reactions. So he said that you can't even generally get ones that will have the same shape and maintain it with any kind of probability. So yeah, that looks really difficult because the number of atoms in the Milky Way is only about 10 to the 65th. Okay, what about finding a self-replicating RNA? So there's been a lot of work towards, looking for RNA, what they call ribozymes. There are some enzymatic properties that RNA sometimes has. So it could be it could be one of the most useful molecule types on the early Earth because it can do some of the things proteins can do, but obviously it also stores information. As most of y'all probably remember from high school, even if you're not taking it here, RNA is what gets, you start with your DNA and it's transcribed to RNA, and then that's used to translate into proteins by the ribosomes. So there's been this big emphasis towards seeing if, can we find out what RNA could be produced on the early Earth, and if we could somehow get it to replicate itself. And there's been some kind of overstated claims here, but basically it hasn't been able to be done to the degree that would make a difference in origin of life at this point. They found no replicator with sufficient fidelity to date, especially with a way of forming it. There's definitely some ways of getting some RNA on the early Earth, but you don't get the right-handed ribose backbone to the molecule that you would need. You get a mixture of of the right and left-handed, so that doesn't really help you. And it's a very fragile molecule. And so even these researchers admit, like, okay, yeah, if we were to find something that could cause replication, you need something probably hundreds of nucleotides long. And so how are you going to get the right sequence of nucleotides through some kind of random search process if it's that improbable of a sequence? All right. One of the main researchers I found interesting on this topic of the RNA world is a guy named Eugene Koonin. He grew up in the former Soviet Union in sort of an atheistic environment, came to the US and has become a prominent origin of life researcher and biologist. And he's pretty honest about some of the problems. But what he actually turns to solve the problems is the multiverse. So he says, well, wait a minute. Modern cosmology says we might have all these other universes out there, and maybe that gives us more chances. Because he recognizes, he thinks the probability that you would get life in this universe is just so low that he wants to appeal to broader resources, for which I should point out we have no evidence for any universe other than our own. And so this kind of shows a level of desperation that you might be led to, even if you're a person who believes in naturalism and is an expert in the field, if you're going to say that the most probable way we got life here was through something as improbable as the multiverse. So he did a computation in this article, which I could point you all to it if you want to see it. It's interesting too in that it's an open access article, and so you can see all the reviewers' comments about the article online as well. And so he had trouble getting it published, even though, as I said, he's an atheist, he's a skeptic. They thought it was too much of an intelligent design sounding. They thought it would give comfort to the enemy, so to speak. And so he kind of really had to argue and convince them that no, I don't think, I'm appealing to something different here. But he made this probability calculation in which he assumed that 1% of every planet in the whole universe was RNA. Now we've already talked about it's difficult to form RNA, and you need the right-handed sugar backbone on the molecule. And so he admits, like, that, okay, yeah, that's probably an overstatement. But his point actually is to show how hard it is and that you need to appeal to new cosmological theories in order to explain the origin of life. That's the whole point of his article. And so he does these calculations that he claims are like, hey, it's probably worse than this, but here's what I come up with. And he put it at 1 to 10 to the 1018th power. Because what he thinks is most probable, more probable than an RNA world with some kind of slow chemical evolutionary process, eventually leading to the first cell, he rather argues that we must have just gotten incredibly lucky, because for life to really take off and evolve, you've got to have something more sophisticated. It harkens back to the minimal complexity of a cell that we talked about on the first slide. You got to have all these genes, you got to have this self-replication mechanism, you got to have things like ribosomes that can make proteins. All known life is protein-based. We don't have any evidence of RNA-based life anyway. So he thinks that's more probable than the RNA world scenarios. There's other researchers that wouldn't be as sort of pessimistic about it, but other ones, again, these are not like Christians, not intelligent design people, but just honest chemists talking about the probabilities faced by a naturalistic origin of life. Christian Schwab puts it about 1 in 10 to the 300 power, which is still like, that's just ridiculously low, right? So I'd be happy to accept that and say that if that's the probability against a naturalistic origin of life, then we're on pretty firm ground arguing against it. Francis Crick, he was the one who sort of discovered he elucidated the structure of DNA. Its helical structure, won the Nobel Prize for it with Watson. So you guys, I know, have heard of him. But I have a book on my shelf where he talks about how he doesn't think life could have risen on Earth because it's just so incredible and improbable. And so he thinks it was seeded by aliens to get here. But the problem with that is you still need some way to get the aliens. And the things we know about Earth, right, it seems like Earth itself is way more habitable than other planets that we know of in the universe. It's got a lot special. And so it's hard to envision how that necessarily helps your probability a whole lot, just to kind of punt to another planet. But at least he wasn't going to the multiverse like Kunin. So, I mean, actually, there have been scientists who have looked at, well, what's the probability of getting, now for Crick, he actually believed in what they call directed panspermia, where the aliens intentionally seed life throughout the universe. Others have said, no, there might have been life somewhere else, and some rocks got thrown off the planet and transported to our solar system. But that also has come under scrutiny by just scientists looking at the odds of getting rocks from other solar. I mean, if you want to say it came from this solar system, that's a lot more probable, right? Especially in the ancient history of Earth, we did get a lot of debris from other parts of the solar system. No problem there. But again, our Earth is way better suited for life than anywhere else in the solar system. So that's not really helping you. If you appeal to other solar systems, that's where it gets real improbable. These solar systems are spread out more than you think. Actually, I'm sure you'll know this, Seth, but like Andromeda Galaxy and the Milky Way are going to collide someday, but almost none of the stars are expected to hit each other. It's just, things are so spread out, it's not like you think. So it's really hard to get a rock from another planet outside of our solar system. The study said it was a 1 in a million kind of thing. Now, one of the origin of life researchers we quoted before, Hubert Yockey, he says, well, wait a minute, I just want to say life is there. It's just sort of a given. I'm going to treat it as an axiom in math, right? But what's the problem with that if you're a naturalist?
It didn't always exist.
Yeah, I mean, it'd be one thing if you knew we lived in an eternal universe or something, but we know for sure that there was a time in our universe where there was no life. I mean, just even going beyond our Earth, which we also know, that just seems really bizarre that someone would even say that to me. But okay, beyond this, there's also a significant class of problems related to the thermodynamics. Almost all of these kind of chemical reactions that life relies on are thermodynamically disfavored. Now, it could have been the case, right, that these things just tended to happen spontaneously, which is really what you mean by thermodynamically favored. They're not hard to make happen. They happen real naturally. But it turns out that's why life needs these like complicated molecular machines and all these things to overcome the thermodynamics inherent. It's why when things die, things just decay, right, in our world. Life will tend to die unless it's supported very carefully with food and nutrients and all these kind of things. So Morowitz actually did some computations one time just looking at the chemical bonding energy in forming one E. coli bacteria. And he said it's equivalent to having your bathwater spontaneously increase to 360 degrees Celsius. So there's a lot of added energy you need to put into things to form life. There's also issues with, for example, assembling amino acids in the presence of water, which, I mean, some people try to, we talked about the organic soup and trying to form things in these ancient puddles, but actually the thermodynamics works against you in those kind of scenarios. They're more likely to break down the proteins than they are to have the amino acids form proteins in any kind of aqueous solution. You also have problems with the dilution, right? If you're in the ocean or something like that, at least, things are just so dilute, it doesn't help you towards the kind of things life needs to do. Same problem with the nucleic acids. Now, one of the other big problems with the origin of life is you have one of the ultimate chicken and egg kind of problems, which came first. Because we know that, at least in present day organisms, we need protein to make DNA. DNA is transcribed to RNA, and we need RNA to make proteins. You just have sort of this vicious cycle where, the ribosome itself that makes proteins, it does so, it's made of RNA itself, and it's made of proteins. And so how do you get that the first time? And some, like Craig Venter, have done a lot of experiments trying to come up with other ways of making ribosomes that can make proteins. And they really struggled to simplify it. It's like 50 proteins and long sequences of RNA. A lot of information in that highly complex molecular machine found in every organism. Now it is different in bacteria and non-bacteria, but it's very complex in either one. So these interdependent systems exist in every cell of every organism, and so they pose a challenge to any kind of naturalistic origin theory. And again, I'm not saying it's impossible to come up with a solution, but rather pointing out that everything we know about science right now is showing that there's not a plausible naturalistic story of origins for the first cell on the early Earth. Yeah, as you all know, the DNA has the blueprints, and you've got to have so many in those ribosomes. And you need, it's just multi-directional. You need everything for everything, sort of, at some level. Yeah, possibly you could start off simpler than that and add on things, but it makes it very difficult. And of course, all of this had to form through unguided natural processes, and you would really need it within a membrane, right? You've got to protect that genetic information, especially if it's RNA, by the way, which is very fragile. You need to be able to protect that in the membrane. And so the cell also has to take in nutrients from the outside. A lot of times that's your proton gradient. We could talk about ATP synthase some, but I want to get into the RNA world first. So yeah, sometimes people posit the RNA world as a way to work around this circular dependency. And that would potentially get rid of, maybe you don't need DNA. You could have started with RNA and added DNA. That's possible. But RNA has some additional issues because of various reasons that we'll talk about. So here's kind of the picture of the RNA world. You might have started off with something like just RNA. So RNA is serving also the function of the proteins, the enzymes. And then you could add proteins later, and then you could add DNA after that. That's one of the ways they posit this. But there was an interesting article called The RNA World Hypothesis, the worst theory of the early evolution of life, except for all the others. So even the advocates sometimes make some frank admissions about how improbable some of this seems. Some of the shortcomings of the RNA world, there was actually a recent article from Nature magazine as well that was pretty frank about origin of life in general and all the different things that are overhyped and not really supported by science. So RNA, it is very difficult to have arisen prebiotically, but I won't say that we didn't have some RNA on the early Earth, just that it's going to be very minimal. And nothing prebiotically has been shown that would give you just the right-handed form that you need. And one of the key issues as well is that it's inherently unstable. As Jim Tour likes to talk about, like if you order RNA, if any of you are working in a biology lab or something, then you need some RNA. They tell you to store it at a negative 112 degrees Fahrenheit. And you have to have these stabilizers and stuff with that. So it really wouldn't, if you had a few cases where you're getting lucky and forming RNA molecules, it's not likely to survive very long at any kind of room temperature or Earth, I could say Earth temperatures for too long. So it makes it difficult for it to really build up. And you also need concentrations. We'll talk later about interference, other molecules that could react with it as well. But if you want to build long chains of RNA nucleotides, then obviously you don't want anything else mixing in with that. So this is one of the reasons why, like Eugene Koonin was saying, I don't think the RNA world happened. We had to go straight to the protein world, as improbable as that seems. He thinks it's more likely than the alternatives. So yeah, we've already talked a little bit about the minimal length. Before you could do some kind of replicator, it's thought you would need something on the order of like almost 200, say 190 nucleotides in length. So you got to get just the right sequence of those, where each one, is the 4 choices for the different letters in order to get something which could have this kind of property. And even that, they really haven't shown to have a reliable mechanism for replicating. The other problem with RNA is the ability for it to serve as like an enzyme, basically, is very minimal. So yes, it can do some things that proteins do to help speed up reactions, but it's very minimal. It's not a full replacement at all for what proteins do in cells. But let's talk about these interference I mentioned a minute ago. So for life, right, you want to form these polymers, these strings of monomers. They're either talking about RNA nucleotides or amino acids. You got to chain together very long versions of these to do useful things. And the problem is that it's real easy for other kinds of molecules to latch on, to those chains, so that it's no longer just a sequence of RNA nucleotides or amino acids, and that would affect the shape, and therefore it's not going to be able to be used as an enzyme in that scenario. And so this is discussed also a lot in the literature. There are a lot of origin of life researchers that will look at experiments done by other origin of life researchers and just say this is so unrealistic, it's not simulating anything that could have possibly happened in the early Earth. Jim Tour, who I mentioned from Rice, is especially prominent at pointing out these kind of flaws, but he's far from alone in that. And even some of the ones that claim, there's a guy named Steve Benner, for example, he was complaining about other people's experiments, and he claimed to have a hands-off experiment. But when Jim Tour studied the details of it, he saw that he actually purchased RNA from somewhere else, and he saw that if you look at the details of his experiment, it's very clear that this kind of thing required too much investigator interference to have happened on an early Earth kind of setting. So that's a big problem, and a lot of that is related to blocking out the side chains to prevent these interference. There's a lot that goes on in our cells that life does through these molecular machines that avoid these problems. But there's an enormous amount of complexity that you had to have to get the molecular machine before you could do that. And those are simply not available on the early Earth. For example, amino acids are actually more likely to bind to other organic molecules such as sugar than to each other. So you can see how that'd be a big problem if you want to form a protein of, say, 150 amino acids, which would be one of the shorter ones that there is. Other molecules disrupt the RNA backbone, cause termination, lead to side branches. Any of those would be fatal to replication issues. There's a number of these kinds of molecules that cause RNA breakdown. And some people talk about, maybe we can posit mineral surfaces as helping origin of life, but that leads to its own set of problems, because RNA will attach to the mineral surface and just get stuck there. It doesn't really get into membranes floating around. And remember, you've got to get all of the right ingredients into that membrane. Robert Shapiro is he's one of the really strong, he's passed away now, but there was one article. He's Harvard educated. He studied under a Nobel Prize laureate who was an expert in nucleic acids. And he was a very strong, outspoken critic of these kind of RNA world scenarios. And that was his area of expertise, was RNA. Now, he posited like a metabolism first scenario, but others who knew more about that argued that that was like pigs can fly chemistry. So it's kind of interesting. But he said the kinds of things that would be required for an RNA world origin of life scenario are like imagining a golf ball being played below par through an 18-hole course solely by natural forces. He's saying, you could posit lightning struck the ball and propelled it over here. Nothing would actually violate the laws of physics, but it's all so improbable to string together that it doesn't make any sense. Another good person to consult would be George Whitesides of Harvard. Charisse, I don't know if any of y'all know her. She came to our group for a while. You probably remember her, Brian. So she's no longer at UTD. She was a researcher here for a while, but I saw her the other day when Casey was in town. But anyway, she studied some under this guy when she was at Harvard. And Whitesides, I looked it up. He was actually like the #1 chemist in 2011 in the whole world. If you looked at the metric where they evaluate your publications based on how often they're cited, which is a good criteria that a lot of scientists use to kind of see how much are you contributing to the field? Not just how much do you publish, but how much do people cite your work? And this is what he had to say about Origin of Life.
So I showed you this before, which is we know how to make chicken soup. The question is, can you make chicken soup with chicken? And that, we don't know how to do that, but it's a little bit even worse than that, because it's really this. So suppose you have something of this kind, which is, I think, what many people would argue is the plausible cradle for life. And then you pour the chicken soup into it, and out of it comes something like that. And it seems very hard to understand how that can happen. How do you go from a system that's disordered, dilute, contains all kinds of other stuff, mostly other stuff, how do you go from that spontaneously, not only to a more ordered state, but also to a state that has the characteristic that, depending upon how you want to phrase it, it's self-evolving or self-assembling. We know how to do that once you get to life, but we don't know how to do it at the very beginning, and that's the reason.
All right, yeah, just a short clip there to kind of show you, and if he doesn't know how to do it, I don't know how any other chemist would, right? It's like this is a big problem, and I think he gave a pretty good summary there of, it's worse than getting chicken from a chicken soup because you got all this other stuff mixed in the soup, right, Jeremiah?
Is he an atheist or a Christian?
I don't actually know exactly. If he's a Christian, he's not one of those outspoken ones or whatever that, so.
Yeah, no, I was wondering if maybe like this realization made him a Christian or something.
Oh, I don't know. I don't think he was willing to. I think, as far as I know, he was more of a naturalist, and he was probably still looking. He had a long history of success of finding challenging problems that no one thought they could solve and solving them, but he certainly never did this one. I don't know that he really specialized that much in Origin of Life, but yeah, I think he was just more of trying to say, look, this is a huge problem. No one has any clue how it could have plausibly happened, and here's why, but yeah. He wasn't like the guy, Smalley, that I mentioned at the beginning. All right, now again, we could play the game of, let's assume all the challenges so far have been met. We've got the building blocks. We've got, the handedness of all the molecules is right, so we can chain them together. We've worked around the problems of the interference. All that's solved. Can we assemble it? That's a big challenge, even if you have all the right ingredients, it turns out, because people have actually done these experiments. They're affectionately called Humpty Dumpty experiments. Can you put Humpty Dumpty back together again? Poke a cell in a saline solution and see if it can close the membrane and be alive again. And the consistent result from scientific experiments is no, you've killed Humpty. So that's pretty interesting, right? When you combine that and you think about the complexity of a cell, how many parts, even if this already has the organelles and everything in it, right? And yet you can't revive the organism once it's punctured. All right, now one of the interesting things as well is that if you go back in time, people have been talking about this since Darwin. He barely alluded to it in a letter, thinking maybe there's some warm pond where life originated, but they didn't know enough for him to even do much speculation. But if you advance the clock a little beyond that, people started thinking that we probably had a really long time for life to originate, because most of our oldest fossils of animals start around the Cambrian, 500-something million years ago. The Earth is now known at least to be 4.54 billion years old. And so, that sounds like a lot of time, right? But actually, the more we've learned, the more this has shrunk. We know that the Earth was in a very uninhabitable state until about 3.8 billion years ago due to the late heavy bombardment. If you look at the moon and you see all those craters on the moon, most of them actually were from that era of about 3.9, 3.8 billion years ago, or some a little bit older than that. But that's when apparently there was a lot of debris in our solar system that was just pounding the various planets and causing these huge craters. And so most people think that there's no way, or even if you had life before that, you'd have to start over. You would have sort of vaporization of the oceans and these really radical things happening in that kind of time frame. And yet, the more we've looked, we have found, yeah, as I mentioned, there was a time where the oldest fossils they thought were much more recent, but we now know that we have some very old fossils. I'll show more details in a minute on that. But as this Walton guy summarizes, it seems now that life originated virtually simultaneously with the formation of the crust of the Earth, once that was sort of finalized. Now that's in a geologic kind of time scale, so they're really talking about, it's only 100 million years or something. But that's a granularity where you can even measure it, going back that far. So they're saying it was probably more like 100 million years than like multiple billion years. But that's a pretty significant decrease that hurts your chances if you're already talking about something improbable, right? And we actually do have, as shown here on the right, this is Shark Bay, Australia, we have these stromatolites. These are mats of bacterial cells that they actually are very complicated. They have a plant-like photosynthesis. And so we have these going back as our oldest fossils, which itself is interesting. How do you get something so complicated so quickly? It's actually doing photosynthesis. And these fossils look just like the modern day versions, as Lynn Margulis pointed out here. So this is a slide that Casey Luskin sent me as we're talking that has some additional information relevant to this. Him being a geologist and also studying biological stuff through master's degrees and so forth. He's summarized some of the information here. This late heavy bombardment ended, as I mentioned, 3.8 billion years ago. And again, this boils off the ocean and sterilizes the Earth, according to origin of life researchers and geologists themselves. So even if you had life, you'd have to start over. And yet we have the first evidence of life. We have some suggestions, some debatable fossils going back into the late heavy bombardment even, but it's much more hard to say for sure. It's like, is that really a fossil or not? But I think we have more strongly affirmed are microfossils from Canada, which can go back as far as 3.77 billion years ago. And we have chemical signatures going back into that same time frame as well. So life prefers carbon-12 over the heavier isotopes. And so if you're looking at rocks and fossils and you find a carbon-12 versus the heavier carbon-13 or carbon-14, significantly, and the carbon-14 will radioactively decay so quick that on older rocks it's irrelevant. But you can tell, like, this looks like it was alive because it's almost all carbon-12. So it would be as though the plant or whatever organism is pulling in carbon from the environment, but only the carbon-12 version. So that's a pretty strong thing as well. But the bottom line is you have multiple lines of evidence for life originating very early. And so your time frame for all these improbable things to happen is actually somewhat short. Now, it might still sound like a lot of time, but when you're talking about these kind of improbabilities, that's not near the time you would need probabilistically. And just kind of summarizing that as well. For sake of time, we'll fly through there. And then one last, the 12th point here, is to look at the fact that origin of life researchers argue that this has to happen more than once. Okay, so even after the late heavy bombardment, not just the starting over aspect, they think that it's very probable the first cell would be very fragile and die off before it could replicate enough to still be around, right? So they think in some articles like PNAS, that's Proceedings of the National Academy of Sciences, very prominent science journal, they think there's probably about 10 of these kind of origin of life events before you had one that stuck. So you got to do this 10 times in a shorter amount of time than you think. All right, so the other thing that's kind of interesting is there also is a group of biologists moving away from common ancestry. That's still rare among biologists, but some prominent folks, and even as seen here in this New Scientist article, Darwin was wrong, cutting down the tree of life. Some people think there had to be multiple origin of life events just to explain biology. So even among ones that survive to the present day, you might need more than one. Now that's more debatable, but it's pretty interesting. Craig Venter, he's the one that did that synthetic cell I referenced earlier. He says, I think we may have thousands of recent common ancestors and they're not necessarily so common. He would do these things where they go out to the ocean and just scoop out water and analyze the organisms through their kind of DNA analysis. And he saw so much variation, they couldn't build trees that would really explain it. And he thinks it looks like there might have been multiple origins. So that's just another minor problem. But if there were multiple origins, note that that also goes against some of the arguments by those such as Richard Dawkins, who points out that if you have the same genetic code with minor exceptions, then he thinks it's almost unimaginably improbable that different organisms, if they didn't descend from common ancestry, would use the same genetic code mapping, that is from the RNA codons into amino acids. That kind of mapping is pretty consistent across all life forms, so it's surprising that you would have that if you did have multiple origins. So in summary, we've talked about the extreme complexity of the simplest possible cell, the fact that there's no evidence for organic soup on the early Earth, that we have barriers, not impossibilities, but it's a lot harder than you might have been led to believe from your biology textbooks to build the necessary building blocks, and especially to get the right-handedness or left-handedness for amino acids. And that even if you had all those right building blocks, you still have to assemble them in very particular orders, which forms kind of an information impediment. And remember too, like if you're going to originate this first cell, you need something like the genetic code to store the information. And that implies that you have a sender and a receiver that are in sync and all that kind of stuff. You've got to get that off the ground from scratch, which is very challenging. And you have some probabilities really working against you, even as naturalistic biologists like Eugene Koonin will admit. And then we talked about the thermodynamics, that most of these organic reactions have to go thermodynamically uphill, and so they need some help in various ways. And you have the chicken egg problems, the interdependence of DNA, RNA, and proteins as being difficult to originate, and the problem of the side reactions that get in the way as you try to assemble these low-level building blocks to form what you need, such as amino acids. And then even if you have all that, you have this assembly problem. It had to happen faster than they used to think, and that probably almost certainly would have had to happen more than once. All right. If you think about these two, I just want to, in summary, point out there's sort of a synergistic effect here. So yeah, if you want to overcome the chicken-egg conundrum, maybe you propose RNA world, but now you're dealing with something that's even less stable than DNA that you're trying to form and build up on an early Earth environment. So that you're making that problem worse. Interference and chirality problems exacerbate the challenge of forming the useful building blocks because of obvious reasons that we've talked about. And all of that makes then the information impediment much worse than it would have been. For example, if amino acids are incredibly abundant, then you have many more chances. The cellular complexity makes the automated assembly challenge worse because what you're trying to build is just so complex. And yeah, the lack of available time, multiple life origin events, it makes everything else, it's just another layer of additional improbability on top of that. So why aren't these problems more widely known? I think Paul Davies summarized it well when he said, many investigators feel uneasy about stating in public that the origin of life is a mystery, even though behind closed doors they freely admit they are baffled. There are two reasons for their unease. First, they feel it opens the door to religious fundamentalists, and second, they worry that a frank admission of ignorance will undermine funding. So there are human elements around in science as well. We're basically out of time, so I'm just going to end it right there. But if you have a quick question, we could take that now, or I'll just hang around at the end. Or if you want to go to Sonic tonight, I'll be there, and we can chat about any of this in detail if you're interested. But all right, go ahead, Landon. One quick question.
The easy solution to all of this from the materialist standpoint is you just rule out God as being impossible at the beginning.
Yeah.
And so therefore, however improbable this may be, if you've ruled out the impossible, this has to be the solution.
Yes. The obvious problem with that is how can you be so sure there's no God to make that assumption and make it so strongly as to basically then you're admitting like I'm not looking at evidence at all. I'm not evaluating anything. I've already decided, which is a very dangerous stance to take, I think. But you're right. It seems to be the stance that some do take, whether consciously or subconsciously. All right. Well, thank you guys for coming. And yeah, we'll see you next week as we're going to get into a series on the Bible. Y'all have a great week and join us at Sonic.
Thank you, Alan.
Probably should have left more time for interspersed Q&A there, but